Indian hedgehog cancer, that's another one of them, and echidna hedgehog are the three vertebrate hedgehogs.
You had a fox later on.
Okay, any other questions just in general? Okay, sort of, it's a little bit of a disadvantage to have a review so quote unquote early, because maybe some of you haven't really gone into depth and figured out what you don't know yet, but we're booking along here, so try as much as you can to keep up day to day, and I think that will help a lot.
You said lecture 18 is included too, right?
So check on the syllabus for the exact lectures that are included, but
And that's with the notch delta type.
right. Oh, so lecture 18 with notch delta. Any questions on that one?
I guess you never talked about, you talked about if they took away notch, you had an overexpression of delta, which did all this stuff. But if you took away delta, would it be a similar situation as opposite?
So mutating either notch or delta is going to end up with too many neurons developing. And it's partly because the job of those two proteins at that early stage is to act as a gatekeeper about how many cells will be privileged to enter into the neuron cell fate pathway. And it's really a matter of dampening down the neighbors. So if you break that process by mutating notch or delta, then every cell thinks it's the one, and it keeps on going and makes more proneural genes and becomes a neuron.
So it's not just taking away notch. I thought it's like delta hits notch and then that tells
Yes,
stop
so-
making more of its own delta.
Right, so, but remember that the outcome of notch and delta signaling is to shut down delta.
Yeah.
So if you break either component, you're not shutting down delta, or you're not shutting down basically the continuation of proneural gene expression, so you still get a neural cell fate. So equivalence group. Do we know what an equivalence group is?
A group of cells that are all basically at the same level, or they have the same potential.
Yeah.
Have the same thing.
Very nice. Group of cells with same level, same potential in terms of cell fate. So this is early on in the decision-making process where everybody has a chance. So all the students can apply to vet school, and they have to then go through this process of, we're going to select certain ones that will go forward and certain ones that don't. But initially, everybody has the same chance to apply.
Well, what was that you just said?
What's that called?
So there's an equivalence group, is the name of the cells in the field that have the same potential initially. Okay, and what is this whole paradigm called? Where we're shutting down, we're selecting only a few cells to take one cell fate out of a choice of two.
Lateral inhibition.
Lateral inhibition. Okay, any other questions? All right, I think you're doing well. So email me if you think of other things along the way, and good luck studying.